Hi, welcome to the lecture on adaptation of animals. Now on the second half of these aquatic environments, we are focusing on the ocean. We all know that in the ocean we have plenty of salt. So that means that there is a risk of dehydration if we drink the salt water. But still, we have animals that are isoosmotic. So, in like this squid, it has very high osmotic pressure in the body fluids. It's 3 times higher than in the human body. Whereas in the teleost fish, they are hypoosmotic, so they are pretty much on the same scale of the osmotic pressure as we, and therefore there's a risk of losing water and getting too much salt. Okay, how we can regulate the plasma content, and do they all regulate it? These marine invertebrates are usually isoosmotic and isoionic. Okay, this isoosmotic means that there is as much milliosmoles in the blood as in the environment, so the water is quite much in the equilibrium. Then this isoionic means that the concentration of different ions, they can be either positive or negative, is similar. But it doesn't mean that there is as much sodium, as much chloride, so the concentration of different ions can be still different. And there is also some species-dependent variation. In this table, we have plenty of different kind of marine animals, from the mussel all the way till the fish. And then there are different ions that have been tested in the body fluids. And then on the right-hand side, we can see I have added some of these ion concentrations. So how much is the total concentration of different ions? And we can see that in all cases, it's about 1100, as well as the ion concentration in the seawater. So they are isoionic. But there are still some species-dependent variation. Like in the hagfish, there is a little bit less potassium, whereas in squid there is much more. And in the calcium, there is a low calcium in the hagfish, but much higher in the crab. And in the magnesium, it's also low in the hagfish. So they are all having some regulation. So they are uptaking different ions, not all ions, and they are excreting some ions more than others. And this hagfish is important because it's the only vertebrate which has the isoosmotic blood fluids. So all the teleost and all the elasmobranch like sharks, etc., they are having different osmotic pressure inside the body than in the ocean. Okay, these marine fish are usually hyperosmotic, so they have a little bit higher osmotic pressure than in the freshwater fish. And in this picture, you can see that, they were testing with the Brazilian coast on collecting different animals and taking the blood samples. And I've been just calculating the average. And you can see that in the ocean, the osmotic pressure is a little bit higher than in the freshwater fish. But the difference is not as steep as they would be the osmotic conformers. So with this black line, it is demonstrate that in the theory if they would not regulate the osmotic concentration, osmotic pressure, then the osmotic pressure would be much lower in fresh water, much higher in the seawater. And why they have this kind of regulation is actually, it's been speculated that actually all these teleosts have had a background with fresh water. So they have much lower osmotic pressure in the sea than it actually would be the ideal. So therefore, they've been first into freshwater and then going back into seawater. And you can see that what is the difference on different ions on the fish versus on the ocean. You can see that, okay, all these are different. And in calcium and magnesium, there's a huge difference between the sea water and the blood. So they are having much less calcium, magnesium, even chloride, etc. So they are not only hyperosmotic, but they are also on several ions, the concentrations are also low. Okay, how they can then maintain to have water in this hyperosmotic body? You can of course drink. And in some cases, these fish are drinking all the time. So they can even take 20% of the body mass with the drinking. But at the same time, they are also taking sodium and chloride. And they are even enhancing the uptake of sodium and chloride in intestine with active transport to get a gradient that now inside the intestine you only have the water. You have already taken the sodium and chloride out. Then of course the water is moving freely with the osmotic to the body. But the problem is now that, okay, you are actually getting much more sodium and chloride than water in this behavior. And then they are using the chills to get rid of the sodium and chloride. In the freshwater animals, we were using the chills to uptake sodium and chloride, but now we are doing it in the opposite direction. And how we can do it is that, okay, we are using pretty much the same mechanisms. So we have sodium, potassium chloride co-transporter, and then we have sodium, potassium ATPase. And with these mechanisms, we can then push out chloride. And in some cases, the sodium is flowing passively. Okay. Then we are then getting rid of the sodium and chloride with the chills, and they still need the kidneys is because of the divalent cations. They have calcium, they have magnesium, but the concentrations of these are much lower. Okay, what it takes for fish to live in the ocean. So the problem is that, okay, you are getting salt and you are losing water because your body is hyperosmotic. And this water loss is not very small. It can even be 8% of the body mass that it's losing the water. And of course, it can drink and eat. And with that, you can get practically all the salt and almost all the water you need. And then, of course, you then have different roads to get rid of the waste products. So you are pushing sodium and chloride in the chills. Then there is quite a lot of water going with the feces and very small amount in the urine. So the difference in the marine teleost and in the freshwater is completely the opposite. So in the freshwater, you are having problem that, okay, you are losing salt, you are getting too much water, but then you are taking actively sodium and chloride in the chills. And therefore, this freshwater animal is much working more better. So it can be using half of the energy of the animal that is in the seawater. But still, we have plenty of fish in the sea. Okay, what about then on marine reptiles and mammals? They are all in the hyperosmotic environment. And they have only slighter osmotic pressure. And we have to remember that, okay, when we have this pulmonary, so when we have the respiration, and so there's the evaporation, and then we lose water. And when we are losing only water, then of course we have more salt in our body. And the problem can also be that in the reptiles, for example, they are not very good in to secrete the concentrated urine. So there might be only 2 times more salt in the urine than in the blood. But we can use salt glands. And these salt glands are there above the eyes. So we have a special gland in these marine reptiles, marine mammals. Sorry, not marine mammals, marine reptiles, marine birds, and etc. And they have these longitudinal loops. So there are secretory tubules in which we are secreting sodium and chloride. And how we are doing it is that, okay, we are secreting it in the bill. Okay, how to make it? So they are secreting highly concentrated fluid with the salt gland, and you can see that, okay, to compare the seawater, this sodium concentration can be even twice as much. So it's very highly concentrated. And therefore, it's actually emitting or excreting more sodium per milliliters of water than when drinking the standard seawater. So therefore, it's the half of the seawater is remaining in the body. So there is no risk for dehydration in these animals. And this excretion mechanism in the birds and turtles is very similar with the marine fish. So there is this sodium, potassium, ATPase. There is this NKCC. So the sodium, potassium, chloride, co-transporter, etc. But then the road actually can be different. So in the turtles, for example, they are actually having tears. And in the birds, it's well characterized that, okay, there is a parasympathetic regulation. So when our nervous system is getting the signal that, okay, now we have too high concentration of salt in the blood, then with the parasympathetic regulation, so acetylcholine is then released, and that causes that okay, now there's a excretion of sodium and chloride, but also with acclimatization. So when these birds are migrating from the freshwater to seawater, so they are then having larger salt gland, etc. So, what about then the elasma branch? We have discussed so far with the teleost and then these reptiles, etc. But the elasma branch, like shark and ray and cascade, they have pretty similar ion concentration with the teleost fish. So okay, here I have the human plasma. So we have maybe 140 millimolar sodium and 100 millimolar chloride, and in this elasma branch, it's not so hugely different. But what we can see that actually the osmotic pressure is completely different. So the blood osmotic pressure is higher than in the seawater. But these ion concentrations are lower. So there's less of salt inside this animal, and the osmotic pressure is high. And this discrepancy is maintained with high organic solute concentrations. They have two compounds. They have urea and then these methylated amino acids that we were discussing earlier, so these TMOAs. And this urea is very exceptional in these non-terrestrial vertebrates. This is not common in any fish. So typically the fish are secreting ammonia that we are going to discuss later. And this urea is very, very high. 300 millimole of anything is very high. And it's actually 100 times higher than in mammals. And what we were discussing earlier, that actually this very high urea concentration is also already affecting on the enzyme activities, and therefore they are using these 3-methylamine oxidase, so to boost the proteins. Okay, but let's look on this shark. So now it's hyperosmotic but hypoionic to the ambient water, and therefore they are gaining the salt by diffusion and gaining water with the osmosis. Sounds very interesting and very logical. And how they do it is that because 40% of the blood osmotic pressure is caused with these organic compounds like the urea. And of course, they are then having the food to get some salt, but water with the prey, but actually they are not drinking at all, because they get with the osmosis much easier the water inside. And of course, then they are regulating on these waste products. So there is the urine that is modestly hyperosmotic. There is plenty of these divalent ions, and then in the rectal there is also some sodium chloride excretion, etc. But is this the optimal way to live in the ocean? Actually, no. Actually, the problem can be that they need as much energy as the teleost fish to support these iron concentration and to because they are now producing these proteins in their plasma. Okay, what about then on these challenges with the salinity changes? So we have animals that are migrating or living in different environments. So some of them are stenohalid. So they can live only on a certain osmotic pressure. Therefore, we have plenty of animals that are never coming in the Baltic Sea because there is still not enough salt there. And then the other opposite is the eurohalin. So we have animals that can survive in different osmotic pressures. And then we have hyperosmotic regulators. So when we have low environmental osmotic pressure, they are trying to maintain the osmotic pressure in their body. And we can see that in carp, for example, teleost fish, it's increasing step by step when having it in the more and more seawater. But then so it keeps it in the 220 to 300 or whatever. But then we have this extreme osmotic pressure that, for example, blue crab is keeping, even when being in almost completely fresh water. Then the other way is that we can have animals that have hypoosmotic regulation. So they are keeping like the ghost crab. It tries to keep the osmotic pressure all the time constant. Like this fiddle crab, it's changing, but it's changing it only slightly, etc. So they are regulating the osmotic pressure, or it's trying to keep it down in high osmotic environment and try to keep it up in low osmotic pressure environment. And of course, one typical example is the migrating fish. We have different trout species, etc., that are living there in the ocean and then coming to freshwater for breeding. And they have responses in the chill proteins when they are transferred to seawater, there's acclimation process, and then before coming to completely fresh water, they are waiting there on the coast, etc., to make this acclimation happen. And the interesting part is that in the blood there is this shift in the osmotic pressure. So they are having different compounds in the blood. And also what happens in the chills and on this whole water balance is that now this sodium chloride transportation is changed. They are either starting or stopping the drinking habit. Then they are modulating what kind of urine they are producing. Are they taking the sodium chloride in the intestine? How much aquaporins they have, because the number of aquaporins is related directly on the flow of water. And one interesting part is in the chills, there is a change in the ion transportation chloride cells with this NKCC. And you can see that when we are moving them from the seawater to freshwater, there's an up-regulating of these of the gene coding this sodium potassium chloride co-transport. So that means that now you have more these proteins in the freshwater, and that means that then you can have more trying to get all the ions from the surrounding to your body. Thank you.